Howdy. If you guys could join me, please give another round of applause for the organizing chair. She has worked around the clock to bring this thing. So, are you guys ready? I'm sorry, that's not going to work for me. I'm sorry. So it was just announced this past week. I don't know if you guys see your emails that A&M is actually #1 research institution in Texas, which means that this is a phenomenal research institution, and a lot of you guys contributed to that. And like I just said, my chair has curated one of the best panels we can have. So I'm gonna need you guys to give me a better welcome in that. Are you guys ready? Yeah. That's what I want to hear. So without further ado, let's get to the first presenter. Joining us from Cornell University is Dr. Larry Smart. He is a renowned plant geneticist dedicated to the science of breeding better crops. Whether it's hemp, hops, or willow, his research focuses on the vital intersection of genomics and disease resistance. He has mentored dozens of future scientists and authored over 70 extension publications to support our growers and processors. We are honored to have him here today to discuss his latest insights into powdery mildew and hemp. Please help me welcome Dr. Larry Smart.
All right. Thank you, Leo. And I also want to thank Merv and Nick and all the other students for organizing this. I'm incredibly honored to be here. I also want to thank Corteva for their long-term support of this program, including our symposium that we host at Cornell. So I have this amazing connection with Texas A&M. I have two former students. There seemed to be a pipeline from Steve Haig's lab to my lab, and Dustin Wilkerson and Savannah Shelnut are Aggies who came to my lab and did a great job. And then a couple of years ago, Brooke Isbell was a summer research scholar in my lab. So, and then one of my students, Brennan Hayden, was one of the invited speakers, I think maybe 3 years ago in this symposium. So I'm really honored to be able to continue that connection between A&M and Cornell and my lab and be here to speak today. So today I'm going to talk about, I tried to fit one of our projects on hemp into this multi-dimensional theme for the symposium, and I think our story on powdery mildew resistance in hemp best fits that. This is a collaboration between my lab and Chris Smart's lab, my wife, who is a plant pathologist at Cornell, and it started with one of my students, George Stack, working with one of Chris's students, Ali Kalla, with Ali doing the plant pathology and George doing the mapping and genetics and genomics. That has continued on with my current student, Lucia Vignale, and Mike Quaid, who is a technician in our employee degree program getting his PhD. And then I'm going to end up with some very new data on gene editing that we're doing together with Mike Peterson. So these first couple of slides I put together probably 4, 5, 6 years ago, just trying to explain hemp and what it means to start a new breeding program from scratch with no resources, very little historical data or any research foundation, no germplasm collection. So I'm not going to give a whole lot of background on hemp because I really want to get into powdery mildew, but hemp is a very diverse crop. We grow it for cannabinoids in a horticultural setting, either in greenhouses or in raised beds with irrigation. We grow it as a row crop, high density for fiber, and that means either the inner hurd or the high quality long bast fibers. And we also grow it as a grain crop. And we just got that John Deere combine, so I'm kind of proud that we're now able to combine hemp grain, which I think has incredible potential for both biofuels and animal feed. And when we look at the goals for hemp breeding, our #1 goal has always been to breed for low or no THC. There is an extreme stigma about THC. It's treated as a contaminant in our animal feed, for example. So right from the outset, we said, let's look at the genetics of cannabinoid production and breed for low or no THC. I think there's tremendous potential for using the protein in the grain, both for animal feed and human food. But we need it's not great in terms of its functional properties as a food ingredient. So that's one of our priorities. And then on the fiber side, we really need to make processing more efficient. So we're looking at both fiber quality, and I know here in Texas, you have fantastic resources for looking at cotton fiber quality. We're so far behind in hemp in that aspect. And then just to get this crop established in the field can be very challenging. It's very finicky, so good seed establishment and early seedling vigor. My student Luis Monserrate, who's shown there, has been working on looking at seed size as a way to promote early crop establishment. And then early on in the hemp, when hemp was legalized, it became very clear that there were not good cultivars to grow at southern latitudes, including in Texas. So we have been doing a lot of work, and I think we now have good cultivars that will grow here at southern latitudes. So the challenges early on were, again, there was no germplasm. And again, I'm looking at these and I'm thinking, oh, over the last 5 years, we've ticked off some of these. We've made quite a bit of progress. So I'm going to talk about how we have been able to access the growing National Germplasm Repository, which is hosted at Cornell in Geneva. So we now have actually quite a good germplasm repository, probably the best in the world right now. We've learned a lot about the genetics of cannabinoid production. I'm not going to talk about that today. And we've actually had some pretty good breakthroughs in genetic mapping resources and an incredible pangenome now that was published last year. Flowering time is still a challenge to us. That's probably one of the things that I'm primarily focused on in my research program. But today we're going to talk about disease resistance and jump right into powdery mildew. Powdery mildew is probably the main disease problem in indoor production because it likes dry, humid, warm climates. It is polycyclic, so you can see in this picture the chains of conidia, which can fly to other plants or other leaves on the same plant and start a new infection during that same season. So it's caused by Golovinomyces ambrosiae. And again, you can obviously see the symptoms there of powdery white lesions on the leaf. We actually don't see it as a huge problem in grain and fiber crops, but it is the major problem in cannabis production and in other indoor protected agriculture cultivation of hemp. So going back to 2019, we were just trying to capture as much germplasm as we could and put it into trials and see what traits were segregating and see where we had opportunities for breeding and genetics and genomics. So here's an example of our CBD cultivar trials that we did, one in Ithaca, one in Geneva, looking at 30 cultivars across those two sites. And again, Ali and George were very perceptive, went out and scored for powdery mildew. So we do get powdery mildew in CBD fields like that, which are very bushy and there's a lot of humidity inside the plant. So it's a good environment for powdery mildew. So quite a range of disease severities there, but one variety that consistently across sites, across years, always was scored as 0 for powdery mildew, FL58. So let's just step back and talk a little bit about, as we're thinking about, well, we see this resistant plant. What is the genetic basis for that? It could be an R gene mediated resistance, which is typically dominant, has a gene fold. Turns out Mendel was right because 25% of those plants were resistant, showed a lack of susceptibility. in both populations. And then we saw a range of susceptibility across the rest. So the next thing was to genotype those about 700 plants. And we turned to Phylos Bioscience, which is a cannabis breeding company and seed company in Portland. And they shared their Illumina array technology with us. And so we got about 2,700 SNPs, built a linkage map. And so there's a nice linkage map. The key thing is that we got one main hit when George mapped AUDPC, which is area under the disease progress curve, or the development of powdery mildew through the season. One big hit on chromosome 1, and a few other minor hits there, which are interesting. But underneath that confidence interval for that QTL peak, it turns out when we align it to the genome, it's pretty tight peak there, and shows, classic example where the homozygote is resistant and the heterozygote and homozygote of the other allele are susceptible, although show a range of susceptibilities. So right under that peak is one of those MLO genes that was predicted by PEPM, MLO1. So there's a very good candidate. Fits the whole story just about perfectly. So we went on and sequenced those two individuals, FL58, which was actually very homozygous inbred individual, and TJ-CBD, which was very heterozygous, basically a hybrid. And lo and behold, when we sequenced the FL58, it had a large insertion in the middle of the MLO1 gene. Looks like a transposable element because we see it in other portions of the genome, and we see it in the pan-genome sequences. And that insert results in a differentially spliced transcript, which has a premature stop codon. So again, it all fits the story. The only little tweak here that doesn't really fit is that we actually were able to recover some of the properly spliced transcript from that allele. So FL58 is homozygous for that insertion in MLO1, but it does make a little tiny bit. So what you're looking at here in this graph is the susceptible line, TJ-CBD, and this is qPCR of the MLO1 gene, either uninoculated or inoculated. And there is an upregulation of MLO1 after inoculation. So that's the first thing we learned. But there is a tiny amount of the wild type transcript that is made. So that's the one little thing that I think we're still hoping to figure out, and maybe has led us to continuing to look at this gene using gene editing. The other thing we've learned is that this MLO1 allele with the insertion is present in some of the other germplasm that we have, but it was heterozygous. So those plants were susceptible to powdery mildew, and were so expressing both versions, both the improperly spliced and the wild type versions. We're able to make a couple of markers that we can use in breeding now, because the next step as a recessive allele would be to integress that into other germplasm, and having a molecular marker would make that much easier to do. We use PACE markers, which are competitive PCR markers, very inexpensive and easy to work with. So we developed a PACE marker for that insertion, but also a gel-based marker that has primers inside that insertion, so that we can diagnose when that insert is present. And both of those work very well and correlated again with the genotypes and phenotypes that we saw in the mapping population. So that's a nice story all wrapped up. Everything fits. We've now got the tools. We've got resistance, broad spectrum resistance to incorporate into other elite germplasm, and it looks like, yeah, it's a clear story of knocking out that MLO1. But there's still some remaining questions. So what were our next steps, and what am I going to talk about next? Does this MLO1 allele confer resistance in other genetic backgrounds? Because we did see those other 4 minor peaks, right? And there is another clade 5 type MLO gene, MLO4. So does that play a role? And we're going to use CRISPR to answer that question. And then the underlying question, there must, what about this PM1 and PM2? There must be other R gene-based resistance in cannabis that we can look for. And then can we stack all of these genes together to get broad resistance to powdery mildew? Okay, so let's go back to this mildew resistance locus O. It's a very interesting gene family, highly conserved across angiosperms. There are a lot of genes, so it's a large gene family in every plant, and we can fit those genes into different clades, which Jacot et al. Here organized. We're interested in terms of powdery mildew resistance in the clade 5 genes, which you see here, which have been very important for disease resistance breeding in other crops. But these genes also play roles in other phenomena in plants, and they seem to be involved in calcium signaling and in touch perception. At least that's what these authors are claiming. So it's involved in symbiotic fungal colonization, root thigmotropism, and in pollen perception during pollination. So pretty interesting proteins playing some vital physiological roles, so that knocking them out may have some deleterious effects. So again, there are 14 MLO genes in Cannabis sativa. Looking at the genome, we're interested in the clade 5 when we're talking about powdery mildew resistance, so MLO1 and MLO4. Just want to point out that MLO4 is much, much larger. It has these huge introns here. So it's about 13 or 14 kb, whereas MLO1 is, again, that FL58 has a big insertion. Okay, so one of the questions we want to think about is, if we knock out these genes, will we see negative pleiotropic effects because of the normal beneficial roles that these MLO genes play? So although many of these knockouts of MLO genes are artificial mutations, they're not natural mutations, when you do knock them out, sometimes you have to knock out multiple MLOs to get powdery mildew resistance. As you see here in cucumber, you have to knock out 3 of the MLOs to get resistance, and then you see deleterious effects. And in Arabidopsis, again, you got to knock out multiple MLOs, and when you do that, you start to see early leaf senescence and cell death. So one of the questions that my student Lou had was, first of all, again, does it work in other genetic backgrounds, and do we see these negative pleiotropic effects of that knockout of MLO1? So again, I told you that we found this allele in some of our other germplasm. So we have, Lou has used those and crossed them with 7 other susceptible lines and generated segregating F2 families for all of those different integressions. And she planted those out in the field. So 21 different F2 families, 60 plants of each family with the parents in a randomized complete block design, which she inoculated 2 times with powdery mildew, and then again, very vigorously rated weekly through the end of the season. Okay, so the answers to those 2 hypotheses. First of all, yes, the MLO1 knockout plants, when they're homozygous, are resistant to powdery mildew. So that would be, here are the 21 different families, And you want to look at the left-hand column in each of these. So that's probably a tissue collection mistake there, that one little dot. But otherwise, across all of these different genetic backgrounds, introgressing the homozygous MLO1 knockout confers resistance to powdery mildew, or my wife would tell me to say a lack of susceptibility. And the heterozygotes and homozygote of the wild type allele are all susceptible. You clearly see that this candida, the candida background is really susceptible to powdery mildew. So that sort of leads us on to the next question. Oh, there are other genes involved in resistance to powdery mildew, and candida is lacking some of those other things. So Lou actually crossed this FL58 MLO1 allele into some backgrounds that already have a fair amount of resistance to powdery mildew. Okay, so that answers that question. The question about deleterious pleiotropic effects, we have this scheme that my student Craig Carlson developed for measuring traits and developing these kite area diagrams. So height, diameter, stem diameter are very good predictors of wet biomass. And we also looked at flowering time. And the answer to this hypothesis is no. We didn't see any negative pleiotropic effects across these 21 F2 families. My main thing that I would look at is wet biomass, and there is no significant correlations between the MLO1 genotype and any growth traits. The only thing that we see is there are some reasonable correlations between genotypes and flowering time, differences in flowering time. And that's not surprising to us because we know that there are known flowering time genes in linkage with that MLO1 locus. So it's not surprising that we would see some co-segregation of differences in flowering time with that MLO1 locus. Otherwise, there are no real biomass effects of introgressing this MLO1 into different backgrounds. Okay, so what about that MLO4? And this is where we worked together with Mike Peterson at University of Wisconsin. And Mike developed some guide RNAs. They're kind of hard to see here, but here in this exon to do some gene edits in both MLO1 and MLO4 simultaneously. And he sent us a bunch of T1 seed from 8 different CRISPR events. This is all using his line, which is called Badger. It's a CBD line. So Chris and I, again, this is the project that Chris and I are doing. It's not a grad student project, so fraught with problems when you let the professor do the research here. But we sowed all these seeds, 25 seeds of each line, and Chris is very good at getting disease, so inoculated those with powdery mildew and scored those, had some good susceptible and resistant controls. And again, I apologize. This is a work in progress by 2 professors, so it's pretty messy. But what we did see is that some of these events, some of these lines were all resistant. Badger is very susceptible, I can say that. So Mike has effectively edited Badger to become resistant to powdery mildew. The next question is, what are those edits? But clearly, 3 of these lines that he sent us were resistant to powdery mildew, and the other lines were segregating. So we've cloned a bunch of these plants, some susceptible, some resistant. I'll just say some of these resistant lines also had some marginal leaf chlorosis, and that trait has been sustained. That phenotype has been sustained through all of our propagation of these lines. I'm trying to get the T2, trying to self these T1 lines to get T2 lines, and then we'll see if that marginal chlorosis continues to segregate with the MLO edits. But that could be an example of some of that, negative effect. Okay, so the green ones are the lines that were all resistant. They're all homozygous for some type of gene editing event in MLO1, whether it's a big insertion, a big deletion, a one base pair insertion, a one base pair deletion. So we think we can explain that resistance because we have knocked out MLO1 through gene editing. This one is susceptible, and the lines that we got are heterozygous for knockout of MLO1. So that makes sense too. The big question that I led into, what about that MLO4? Well, here's a line. It seems to be homozygous wild type at MLO1. Has a one base pair insertion. We think that it's homozygous at MLO4, and it is clearly susceptible to powdery mildew. So right now, our conclusion, and again, messy work in progress, is that MLO4 is not playing a role in susceptibility to powdery mildew or resistance. Okay, so that sort of wraps up where we are in the MLO story. The next thing we want to do is look for resistance genes. And so again, we're very lucky to work right alongside Zach Stansel and Tyler Gordon, who run the National Germplasm Repository for hemp, and my student Lou Vinale, and actually one of Chris's students, Jocelyn Schwartz. We're very fortunate to screen a good portion of the germplasm repository, Lou for powdery mildew, Jocelyn for septoria. And so again, planted all those plants out, did controlled inoculations 2 times, scored weekly for powdery mildew. It's a lot of work. And Lou did a great job. And this is, again, AUDPC. We did it 2 years, 2023. Let me just check. We had 70 entries in 2023. Some of those we carried through into 2024. So we had 98 in 2024. And again, you're seeing here FL58, resistant to powdery mildew. And then a lot of these seed lots are open pollinated, so they're segregating. But in 2024, again, pretty good evidence that there's quite a bit of powdery mildew resistance in the germplasm collection there. And these little dots up here are just scored for incidence. And again, you see some lines where some of the plants in the field trial had 0 incidence of powdery mildew, and a whole bunch of lines maybe had one with a score of 0.5% disease. So there is resistance, and Lou has now collected those resistant plants and cloned them and crossed them with susceptible lines to develop mapping populations. Again, looking at the F1, we see that some of the F1s here are all resistant. So that's a very different result from the previous experiment. Some of them are segregating. One of them, the resistance was due to MLO. So when you make that cross and the plants are heterozygous, everything is susceptible. So we didn't want that. We want these lines that are seem to be heterozygous for a dominant resistance gene. So Lou has now selected those F1 plants and done self-pollinations. This is the way that we can do a whole bunch of pollinations in the greenhouse, is we put plants into a pot, cover it with a bag that excludes pollen exchange. So she's done all that to generate the F2 populations. We're about ready to plant out in the greenhouse 300 plants of her best F2 family, inoculate those indoors. We're actually working with Canon USA, who's going to put some cameras up above that experiment in the greenhouse so we can phenotype it by eye and also phenotype it with Canon's high resolution cameras. We will genotype that with the what we call CanSelect genotyping system that we've developed, and hopefully map some dominant QTLs for resistance genes. So that's next. So I put sort of an open check mark there. I think we're right on the doorstep of identifying those dominant resistance genes. And that will then lead us next to this ability to stack resistance genes along with MLO susceptibility genes and really incorporate durable resistance to powdery mildew in elite cultivars. So that's where we are in the powdery mildew story. Look forward to chatting about the other projects that we have going on hemp in the lab. But I really need to thank all the folks in my lab, our funding sources, both primarily from New York State Ag and Markets, but also we have formed a hemp research consortium with FFAR, and some of this work was funded with matching funds provided by Scouts Corporation. So I will stop there. Thanks again for the invitation, and I'm happy to take any questions.
I'll get us started. You mentioned that one of the most laborious parts of this is doing the screening of susceptible plants out in the field. What sort of differences have you observed, if any, between individual raters, or are there automated phenotyping approaches in place that maybe examine other ways to get that data?
Yeah, so we have a lot of conversations about this over the dinner table. So Chris has built a long career over being able to go out to the field and walk down a row and say 2%, 5%, 8%, 25%. She's a very good rater, has a very well-trained eye across a wide range of diseases. And she can do it very quickly, right? As quick as a robot can drive down that row. So she's a pretty good rater, and she has trained her students to do that very well as well. So when plant breeders then want to start taking pictures or use drones and say, oh, drone is better than you, Chris, or better than your students, yeah, I'm not sure that's true. Yeah.
I wanted to ask about the male sterility, or I mean, are there different male and female plants, and how do you do that in those bags and so forth?
Yeah, so hemp is dioecious, but there are quite a few lines that are monoecious. So breeders have identified a weakness in that dioecious that leads to monoecious. Those plants are, so that's one story that I didn't talk about at all that we've made tremendous progress on in the last 2 years, is identifying the sex determination regions and the sex chromosomes in cannabis, and identifying where those genes have variants that lead to monoecious. But a lot of the cultivation of cannabis is only XX females, but it's pretty easy to flip them and get them to make anthers and make pollen. And all of that pollen only has X chromosomes. So you get what we call feminized populations. Right? But the pollen flies everywhere. So it's very hard. It's not like in wheat breeding where you can plant 3,000 plots and then go harvest the seed and move on. We plant one plot because the pollen can fly for 3 miles. So.
I had a question about the MLO genes, the susceptibility genes.
Yeah.
Do you find that that gene family also causes susceptibility for many other diseases, or is it just powdery mildew?
Yeah, that's a fantastic question. I'll say we're working. Yeah, it's a great question. So hemp is also susceptible to hop powdery mildew, but not. It's very hard to get good hop powdery mildew disease on hemp. I'm also a hop breeder. When we have a whole bunch of, so the hop powdery mildew does not grow well on hemp. The hemp powdery mildew quickly will initiate disease faster than the hop powdery mildew. So that's been a challenging thing when we have both powdery mildews in the greenhouse. to be able to do those experiments, but we would love to know if, for example, does MLO4 confer resistance to hop powdery mildew in hemp?
Thank you.
I'll say we're working in parallel with Dave Gent's lab at Oregon State, and he has a student who has been gene editing the MLO genes in hop. So we could ask the same question in hop. When you knock out those MLO genes, does it confer resistance to hemp powdery mildew, which will also go to hop?
I have one question. So it seemed like you are phenotyping F2s. So is that due to the depression of fertility if you progress the lines any further? And if so, how do you have a trick to actually narrow down those regions, just brute force numbers?
Yeah, we've only gotten to the F2 lines at this point, but yes, there is definitely inbreeding depression in hemp as you advance to advanced generations. You will start to lose fertility, even in the F3, F4 lines. We start to see, morphological variants appearing. So yeah, there's definitely the risk of inbreeding depression. Although I think the future of hemp breeding will be to start to breed inbred lines and try to purge the genome of those deleterious alleles and start to develop true F1 hybrids. Yeah.
Great. Well,
We have time?
please